Hello guys, I'm Dr. Isa Khan and you are watching MedicoTube. Our today's session on the remdesivir and antiviral agent that has been added in the COVID-19 regimen few days before. This drug was discovered many years before and it was also added in the treatment of Ebola infection, but it was not that much effective in the Ebola. So now they added this drug. This is on the trial basis for the COVID-19. So we will see its mechanism of action and its side effects, and we will go on the molecular level that how it affects the COVID-19 virus. So let's see, and please remember to subscribe the channel. So we are going to discuss remdesivir and its effects against the coronavirus. Remdesivir is an antiviral agent. More specifically, it is a prodrug of something we call GS441524. Now, what is a prodrug? Prodrug is an active form of the drug, and when it is administered to the body, it becomes metabolized and into the inactive form of the medication. And the active form of the remdesivir is GS441524. Now what is this number means? What does it indicates? It indicates that it is a adenosine analog. Now what does that mean? Adenosine is a nucleotide base without a phosphate group. These are the building blocks that are used to produce the nucleic acid like RNA and DNA. Now, an analog has the properties that it has the mimic action. It is not the real thing. It looks like the real thing, but it is not. That is what adenosine analog is. It looks like adenosine, but in the real, it is not the same adenosine. Remdesivir was originally produced to treat the Ebola virus infection several years ago, but it was not that much effective against the Ebola infection. Now let's discuss the mechanism of action. Actually, it acts as a nucleotide analog to inhibit the RNA synthesis. Now we are gonna talk about this in more detail. So let's see. When a host cell come in contact with the COVID virus, it infects the cell by different mechanisms. The coronavirus has proteins on the surface. These are called spike or S protein. These proteins allow the binding of ACE2 receptor on the host cell. So once the S protein binds on the ACE2 receptor on the host cell membrane, the host cell membrane invaginates and that invagination process closes over and it brings into the coronavirus into the cell, and we call this is endosomes. This endosomes is brought into the cell, or sometimes it fuses with the lysosomes, and that process is called endocytosis. After that, the corona exit out the endosomes and reveal its viral RNA. This viral RNA is the genetic code of the coronavirus. The coronavirus is a single strain RNA virus. Once the RNA is inside the cell, it hijacks the cell machinery like ribosomes, and they look at this piece of the viral RNA and they make a protein translated into the viral protein. These proteins are called RNA dependent RNA polymerase, or it's called RDRP. As its name shows, it is dependent or on the RNA. It needs RNA to function, and RNA polymerase means that it's make RNA. it actually make more viral RNAs. These RNA are also used to produce proteins like spike protein that coat the viral envelope and activates all these viral proteins. And to collect all these viral pieces together to produce a new virus that then can exit the cell to infect the other cells. Now we know that how COVID can bind, intact and infect the cells. Now let's see how does remdesivir work. So when we administer remdesivir, it enters into the cell, metabolized to its active form GS441524, as we discussed before. It is a adenosine analog. It looks like adenosine, but eventually it is not. Viral RNA is like our oven RNA and DNA. It's makes up by nucleotide bases. Actually, there are 4 nucleotide bases that are used to produce the RNA and DNA, but there is slightly difference between DNA and RNA. In RNA, there are adenosine, guanine, cytosine, uracil. The GS441524 is the adenosine analog. Adenosine is a nucleotide. So what is important here is that it acts like adenosine, but it is not the same as the piece in RNA. RDRPR becomes confused. It thinks that GS441524 is a piece of adenosine that's supposed to be used for making viral RNA. Here is the point. It picks up this piece and incorporates it to the viral RNA strand. What happens is, you can think up the piece here is the piece of GS441524. As it's producing the viral RNA, it adds into the viral RNA this piece. When it adds this wonky piece, is not supposed to be there. So what happens is that this becomes defective. So we get one defective and inactive viral RNA. It is not able to do what it is supposed to do. That's how remdesivir works, by inhibiting the viral RNA production. Now let's discuss about some adverse effect of the remdesivir. The most common side effect of remdesivir is nausea and vomiting, and the second one is transaminitis. Now what is transaminitis? Transaminitis is a term used for the elevation of liver enzymes. So this was all about the remdesivir, and please remember to subscribe the channel and press the bell icon. Thank you for